Our next speaker is Tracy Stepien from the University of Florida, who will talk about optimal control of combination immunotherapy for a virtual murine cohort in a glioblastoma-immune dynamics model. Okay, thank you.
All right, thanks. First I'll start off with just a little overview of glioblastoma or GBM, because I don't think that we've had that just yet. So usually this slide has been covered a million times by now, but glioblastoma, I'm talking about the most common type of primary brain tumor, right? So this is not tumors that have metastasized to the brain, but these are tumors that have started in the brain. Median survival time with treatment is only 15 months. I was looking at this reference right here, and I realized it's kind of old, but this number hasn't changed. All right. So the fact that this number hasn't changed means that we need to do something about standard of care, right? We need to be looking at different treatments to try to extend this. So one treatment that we are looking at is immunotherapy, right? So immunotherapy seems to be working in some cancers, and we're having mixed results for GBM. So immunotherapy with immune checkpoint inhibitor, anti-PD-1, we've had some pretty promising outcome in early phase trials, but by the time it gets to randomized phase 3 clinical trials, not so much anymore. However, one of the labs that I am working with, they have a recent paper where they were looking at a combination treatment of 2 different types of immunotherapies. So this was in mice, but they were looking at anti-PD-1, and they were also looking at this CCR2 antagonist. This is a drug that targets myeloid-derived suppressor cells, right? So these are cells that kind of stop T cells from, doing too much. And this combination treatment was shown to extend the overall survival. So we have the survival curves here on the right. So this was with 2 different types of glioma cells that were implanted in mice. In the black is the control, so no treatment. The red line is for the red line is for the CCR2 antagonist only. The blue line is for the I switched that around. Sorry, the red line is for anti-PD-1. The blue line is for the CCR2 antagonist. And then the green line, right, that is for the combination treatment. And so survival has increased. So this is one of the things that we have been wanting to look at. And so this is a little bit more of the experimental data from the lab. And so this is some imaging data. So we can see that we have kind of the outline of where the tumor is. And so they have these colored markers so they can see where different types of cells are. So in the white here is T cells. These are all T cells with the red and the green. So if there's a spot where red and green show up, that's where the MDSCs are. And then in the blue, that is all viable cells. So we make the assumption that all of the cancer cells are the blue subtracting out the green, the red, and the white. And so with this data, we wanted to then try to build a mathematical model and try to understand the interactions between the cancer cells, these 2 different types of immune cells, and then can we look at a combination treatment and see, like, can we do in silico clinical trials to see what we might need to do to improve survival. All right, so this is our model equations with treatment. So for our tumor cells, we assume logistic growth. We assume that there's a killing by T cells. With the T cells, there's just a natural activation of them and a stimulation when there are cancer cells there. We have this term right here that represents the inhibition by the PD-1, PD-L1 complex. So we had a talk yesterday that kind of introduced that. And then we also have inhibition by MDSCs, right? Because the MDSCs kind of will limit the how much the T cells do, and then a natural cell death. And then for the myeloid-derived suppressor cells, we have stimulation by chemokines. So the cancer cells, they send out chemical signaling that recruit MDSCs to the brain. Right, when there is no cancer, you will not find MDSCs in the brain, but when there are cancer cells, then you have MDSCs that start getting recruited, and then we have a natural cell death. So in the orange are all the different parameters that are in the model, and then you'll notice that we have this function U1 of T and a function U2 of T. So the U1, that is for anti-PD-1, so the application of that drug, and the U2 is for the application of the CCR2 antagonist. And the way that we've implemented it in this model and kind of the way to think about it is that it's really more, it's not exactly the dose of the drug that's given, but rather a reduction in the effect of the different parameters. So this rho is this inhibition rate. So the U1 will reduce the effectiveness of that inhibition. And then for this U2, it's reducing the effect of the cancer cells being able to recruit the MDSCs. So when we started analyzing this model, right, we had done some existence uniqueness. We did parameter sensitivity and identifiability. And with those results, we determined that there were really only 5 parameters that we needed to target and 5 parameters that we needed to look at in order to do like a personalized treatment for any sort of mouse. So those parameters, we got lambda C, which is the tumor growth rate, eta is the T cell kill rate, rho is the inhibition rate by the PD-L1, PD-1 complex, R is the inhibition rate by MDSCs, and D sub M is the MDSC death rate. And so, right, essentially, we are going to use optimal control to optimize a treatment for each individual mouse, but we created 2 different virtual murine cohorts in order to, right, so not just look at one mouse, but to try to think about a whole population of different mice. So the first cohort that we did was just we used some Latin hypercube sampling for our parameter values from our literature searches and from parameter analysis we had done. We had lower upper bounds for parameter values, so we just did Latin hypercube on, again, these 5. And then the other thing that we did is, so in a previous treatment-free model, we had done approximate Bayesian computation so that we could get probability distributions for each parameter. So instead of just having a uniform distribution, we had ideas about, you know, if it's a normal distribution or something like that. And so then we sampled from those probability distributions to get our other cohort. And so for the optimal control problem, for the objective function, just so that you can see it, we minimized our tumor burden and the toxicity of the immunotherapies. I can tell you more about the form and the weights and all that sort of thing if you are interested. But essentially that is subject to our ODE system with initial conditions from the experimental data. We have the ending time point free. And then again, from the literature, we look to see kind of what the maximum allowed dosages for the 2 different drugs would be. And so this is a result for one typical mouse, right? So what we would do is we would allow first pre-treatment. So like we would have the tumor being implanted and allow it to grow. And then treatment would turn on sometime after day 7. And that would be the phase where you would have optimal control. So we used software in order, actually, I think that was on the previous slide. So we have software that we used in order to GPOPS2 to give us what the optimal treatment should be, right? So the optimal application of the anti-PD-1 and the CCR2 antagonist. And then the other thing we were interested in is, okay, so you have this treatment happen, but let's allow the patient to keep going to see what happens after that treatment. So after that optimal treatment for that patient, let it the simulation keep running. And so this is an example where the patient then the tumor got too large. So we assumed that that would be death. And so from these results, right, we took our treatment regimen that came out from this analysis, we compared it to the experimental treatment regimen that was used for that original experimental data. And we determined that you should start later after the tumor implantation, but you should have it last longer. So this is something that we would like to test experimentally, but we haven't done just yet. And so we also looked at survival analysis with our 2 different cohorts. So we have 22 days and 55 days. That corresponds in mice years to 2 years and 5 years, human years. And so we can see that with kind of our uniformly sampled cohort that, within 2 years, right, we have 60% disease-free and 50% after 5 years. So that seems to be pretty good when GBM is not that successful. But right, this is the uniform cohort. So when I look at kind of the GBM specific parameters. So taking the second cohort and looking at the specific parameters. So our survival does decrease. So we have 40% and then 30%, but that is still much better than what we are seeing clinically right now. And so we just kind of have these survival curves up here. So this work is showing that this experimental or the treatment regimen is promising. And of course, right, we would need to continue and actually test that. Kind of this are some summary of the other things that we found from our parameter analysis. Our high tumor growth rates and low T cell kill rates, if you have that, treatment failure is more likely. This was mentioned in the projects, right? This would be a case that potentially impossible to amplify the immune response enough in order to do anything. And then high inhibition rates by PD-L1, PD-1, and by MDSCs requires a more aggressive treatment. And that seems reasonable. But then if you have increased immune suppression, then you're going to have treatment failure anyway. And so then kind of like out of all of these parameters, right, if there's a parameter that you need to target as you're thinking about treatment, it does seem to be the MDSC death rate. That seemed to be a much better indicator of long-term survival than either of the immune suppression parameters. In terms of future directions, right, so that image that I had was a nice spatial image, and an ODE model completely ignores that. We are currently building an agent-based model. We're using PhysiCell to explore the spatial distribution and the correlation between where those cells are located. All right, and this is my group. So Hannah Anderson, she was the main person on this project, and Jillian Carr is working on the agent-based model. This is the rest of my group, and this paper, it's on bioRxiv, and as of yesterday, accepted into Journal of Theoretical Biology. So you'll see that soon. Thanks.
Okay, thank you, Tracy. Are there any questions? A quick question. Right.